Thank you for the opportunity that you're giving me to present my work. My name is Francesco, and I'm a postdoctoral fellow at Stanford School of Medicine. And I'm going to tell you a little bit about my work on deconvolution. So the premise behind what I'm going to show you is that oftentimes we work with heterogeneous samples that have a cellular composition that is mixed, and oftentimes we want to understand what the proportions of those cell types are, particularly linked to pathology. For example, you want to understand whether there are cell type changes, if you have a disease, if it's prognostic, diagnostic, or a potential therapeutic target. So you can do this experimentally, especially in the case of blood, using something like flow cytometry or mass cytometry. And that's something that Aaron Newman presented in his nice talk yesterday. And as he said, it's a great approach, but it has its limitation. It can be nicely complemented by cell mixture deconvolution. For those of you that are not familiar with it, cell mixture deconvolution is a computational strategy. Basically, you start from the same sample, you do bulk gene expression analysis, and then deconvolution allows you to go from gene expression data of the bulk to cell proportion estimates. Think of it as in silico flow cytometry. So the way you do this is very simple. You start from the bulk gene expression data, and what deconvolution does, it compares this data to a reference expression matrix called the basis matrix. This matrix has expression profiles for the cell subsets that you expect to find in the tissue that you're deconvolving. So for example, if you're looking at blood, you want to have a basis matrix that has immune cells. And then deconvolution will return these proportions. Now, deconvolution is a very established field. It's been around for like about a decade. And there's a lot of approaches that are currently available. CyberSurf being one of the latest ones. And the thing that I'd like you to take away from this table is that all these approaches, although they're different, they have two commonalities, two common ingredients. First, they all rely on an algorithm, which is the mathematical engine for deconvolution. And secondly, they require the basis matrix, which is the matrix that I was telling you about earlier. If you have these two components, you can go crazy, go download every publicly available data set and deconvolve them and basically have new biology with a large sample size. This sounds easy, but there's a caveat. When you're looking at publicly available data, it comes at a price, and the price is its enormous variation. On one end, you have biological variation. You're looking at samples that have different diseases, and you compare them with controls, healthy controls. But on the other end, there's also a lot of technical heterogeneity. In Geo, you can find microarrays from over 42 different platforms, and there's a growing body of RNA-seq data that get injected in the database, almost daily. And the key question here is, how does heterogeneity affect the accuracy of deconvolution? So we didn't know the answer when we started, and when we decided to answer it starting from the technical point of view, so looking at the effect of platform. To do this, we deconvolved over 1,000 human PBMC samples that were profiled across multiple different microarray platforms. To make things general, we used 4 different algorithms and 2 different blood signature basis matrices. And what we really want to do is look at the accuracy of the deconvolution across our samples and compare one platform against the other. Now, ideally, you want to use a gold standard for this, like flow cytometry, but we didn't have it for all the samples. So instead, we relied on a different metric called goodness of fit. Goodness of fit is basically a correlation between the original expression data and a reconstructed expression vector from the proportions estimates of deconvolution and the basis matrix. If deconvolution did a good job at estimating the proportions, this correlation should be pretty high. That's basically the gist of it. So this is the summary of our results. And as you can see here, these stacked bar plots are organized based on algorithm and basis matrix. Each column in the bar plot represents all the samples that belong to a particular platform, and the colors indicate, how accurate the convolution was. Two things that I'd like to point your attention to. First thing is, if you use the same matrix, no matter what method you use, results look pretty similar. Just look at the rows and compare, different columns within the same row. But on the flip side, if you look at a single column, same method, you compare different matrices, results look quite different. So we computed a quantitative estimate of this by doing pairwise correlation. And what transpires from this is that the basis matrix is, in fact, the major determinant of deconvolution accuracy, not the method. So that's point number one. Point number two, if you look at each individual bar plot, you compare the columns against each other, you will see that they're quite different. There's a lot of variation when it comes to deconvolution accuracy, depending on the platform that was used to generate the sample expression profile. So that's not great. That's exactly the opposite of what we would have in an ideal situation. And we quantify statistically the heterogeneity of these analyses. And in both cases, for both matrices, we observe significant heterogeneity. So this is a problem, and we wanted to solve it. Now, because we noticed that most of the accuracy of the convolution depending on the matrix, and because the basis matrices that we used were actually built from homogeneous expression data, so same platform and only healthy samples, we decided to build a new basis matrix that would hopefully solve this problem. And here I present to you immunostates. Immunostates is different from other matrices because it basically encapsulates a lot of technical and biological heterogeneity. It's been built using over 6,000 expression samples profiled over 42 different platforms. And the samples that went into it came from both healthy subjects and disease-affected individuals. I'm going to skip the details on how I built immunostates, but this is what it looks like in its gory beauty. 20 different cell types, over 312 genes. And for every cell type over there, you can see a cluster of genes that are specifically expressed in that cell type and that cell type alone. When we apply immunostate to the other data set, you can see that overall, the accuracy of the convolution increased systematically, and there's no more significant heterogeneity between different platforms. So effectively, we were able to reduce platform bias to a non-significant level. So that's one side of the coin, the technical heterogeneity. What about biological heterogeneity? Well, we decided to ask whether the disease state of the sample affected the accuracy of the convolution. And to answer this question, we used a publicly available gene expression compendium that collects samples from either healthy or disease-affected individuals. And these samples can be either blood or blood-derived or from a tissue biopsy. And we perform a very simple analysis. So starting from healthy samples, we did the convolution, and we look at the distribution of goodness of fit in blood-derived samples and solid tissue biopsies. And as you can see here, no matter what matrix you use, blood is systematically higher than solid. You expect this. It's by design because you're deconvolving a blood sample using a blood basis matrix. You wouldn't expect anything else. However, when you're looking at disease samples, things are a little bit different. You don't see the same pattern. As a matter of fact, in the previous matrices, you see an overlap between the goodness of fit distribution of blood and solids. So in other words, you lose accuracy. And that is not true for immunostates. That clearly separates the two. So what happened is that the genes that were present in the previous matrices were actually affected by the transcriptional state of the cell due to the disease. Therefore, they were not universal cell markers, whereas in immunostate, that wasn't the case. We extended this across all matrices and methods, and again, we represented this as ROC curves. Healthy samples, doesn't matter what you use, everything works great. When you look at disease samples, immunostates outperform other matrices, and again, independently of the method. So now that we've seen that immunostates was able to deconvolve accurately independent of technical and biological bias, we asked whether that also translated in higher accuracy in the convolution when we had a gold standard. So to do this, we deconvolved data sets for which we had matched flow cytometry data. As you can see here, immunostates produces cell proportion estimates, they're better correlated with measured cell proportions for the same samples compared to other matrices. But also, if you zoom in on two representative cell types, you can see that the estimates are not just correlated, but they're closer to the actual measured value. Whereas in other matrices, you see systematic over and underestimation. Extending this to 5 different data sets for over 400 samples, you can see that independently of data set and method, immunoStates produces cell proportion estimates that are just closer to the measured cell proportions. So with this, I'd just like to summarize my results. We found that cell mixture deconvolution is actually affected significantly by both technical and biological variation. And it depends on the matrix, not the method that you use. So we designed immunoStates, a new basis matrix that reduces both technical and biological bias, and as a result, gives you higher deconvolution accuracy. So with this, I'd like to thank all the people that helped me in this journey, particularly my advisor, Parvesh. And although I'm co-advised by Mark Davis, I basically spend all my time in Parvesh's lab. My colleagues, collaborators, source of funding, and finally, I would like to thank you for your attention. And I'll take any questions. Sure. Let me go back to that slide. Yeah, so basically the idea is you collect the samples that you start with are sorted cell expression profiles for the cells that you care about. We simply collected them, annotated them. We co-normalized together simply by using quantile normalization. And then from this co-normalized expression matrix, we built cell type specific signatures. And this was done in 2 steps, actually. One step was basically looking at the cell types. You can look at them as individual cell types, but also you can look at their families. We first built a signature set for the families to which each cell type belonged to. And then within each family, we built a cell type specific signature that separated subsets within that family. And we found that that design actually did a little bit better than using one against each other in general. And once you have the signatures, and you simply use the data to create the expression matrix, and then you do all the testing. Is it available? Not yet. Pretty soon. We're very close. Very, very close. But thank you for your interest.
Hi. Sorry, I've actually missed the first part of your talk, so yes. The question might be stupid, but. So can you apply this to sort of any type of tissues with the metrics that you already have? Say that I have like synovial biopsies from rheumatoid arthritis patients. Right. We know there is a lot of immune cell infiltrates, right. But we don't really know, what are the cell proportions. Is that something that you think could work with this tool?
Right. So if you're interested in immunoinfiltrates, you should use immunoStates. The thing about the challenge in identifying immunoinfiltrates is just the background that you have from the tissue. But that's something that support vector regression fix. What we find is that if that is not a problem anymore, you should use the matrix that works the best. So this is completely applicable. And given the results that we see, it's probably going to give you a better estimate of those infiltrates.
Sorry, I think they already asked, but so this will be published soon, or can we download this from somewhere? Or
it will be. It's gonna get out soon. We're really working on it. Thank you for your interest. First of all, thank you. I really feel the love, and you'll know. I'll write you an email or something. It's really gonna be out pretty soon. So where should we be checking? We want to send it to bioRxiv first, and then it's going to go for peer review. So you guys can already start, you know, sinking your teeth into it and take a look. Thank you. I really appreciate it. Okay.